You haven't looked at fungi yet. Did you guys look at the flagellar thing?
Mm-hmm.
Okay.
Yeah, the Paratrichus is hard to believe.
The Paratrichus, the cells are very close together.
Okay.
You kind of have, and you see waves of cells. So you kind of have to look and find a region where you can see individual cells, and then you see what looks like just threads all over the place. Those are their flagellar.
Oh, okay.
But you do have to look at them with the oil immersion objective, otherwise you can't see them. Why is that little? So this is a lot of repetition from information that I presented in lecture because we did lecture on fungi. And here we're looking at slides of fungi. So the science or study of fungi, again, is mycology. The root word myces means mushroom. Here you can see a puffball. This is a pretty good size type of fungus. In the Riker mounts that are up here, these things are also puffballs with very long stems. And then you can see the spores coming out of these guys. Something like this would be supported by a lot of filaments that are way down in the soil. So these things were coming up out of sand, but the vegetative mycelium would have been down where there was some moisture. This kind of a structure, this is the reproductive body, and it would also be supported by a vegetative mycelium that's down in the soil. So they're eukaryotic. They're not in the kingdom Plantae, although you can look at various references. For example, if you look at wildflower manuals, every now and then you'll see pictures of mushrooms. It's like, no, mushrooms are not plants. They're fungi. These are puffballs coming up out of the soil in the desert. These are brachyfungi. Those are mushrooms again. And that's a morel. So, plants are generally photoautotrophs. Fungi are never photoautotrophs. They don't have green chlorophyll pigments. They're functionally chemoheterotrophs. A lot of them are saprotrophs. Some of them are parasites. They do absorb nutrients, but again, don't learn the word osmotropic. It's not, it has nothing to do with osmosis. So what fungi do is dump a bunch of digestive enzymes into their environment and then break their nutrients down into small molecules which they can easily obtain through facilitated diffusion. If you think about it, we also dump digestive enzymes outside our cells. It's just we dump our digestive system, we dump our digestive enzymes into our GI system, which is a tube that runs through our body. Think of it as a long, folded, complex donut hole. What's in the hole is not in you. Then again, yes it is. The whole tract is inside. Anyway, we do the same thing fungi do. Fungi can be respiratory and obligately aerobic. They can also be fermentative, which means they're facultatively anaerobic. As far as I know, there are no fungi that are obligately anaerobic. So different ones. Some are respiratory, some are fermentative. Generally, they're mesophiles in terms of temperature range, and they tend to like a pH a little bit more acidic than what bacteria like. So their environments, they often will be growing in environments that are a little more acidic. This particular mushroom that you see, this was a picture taken in Washington, and we were hiking on a trail and watching deer eat these fungi. They were just biting off big chunks of them. So they are definitely a food source. Again, they can be divided into 3 categories based on morphology. The yeasts, which are single cells, the molds, which are filamentous and microscopic, and the fleshy fungi, which are also filamentous but macroscopic. Technically, fleshy fungi aren't microbes. They're not microscopic. This is the yeast type fungus. As I said, they are single-celled organisms. They form colonies that look like bacterial colonies. Usually smooth, shiny surface, circular, low convex, that kind of thing. The body is a mycelium, and whether you're talking about fleshy fungi or mold-type fungi, the mycelium is made up of hyphae. Yeast usually don't form hyphae, although we saw Candida forming what are called pseudohyphae. So the hyphae are the threads, and they come together to form a mycelium. Specialized hyphae can be conidiophores, sporangiophores. I mentioned these in lecture. These are not something I'll ask you on a lab exam. I will ask you for the spores, however, conidiospores and sporangiospores. So originally fungi were classified based on their mode of reproduction, specifically their mode of sexual reproduction. So there were several groups of fungi, and then there were these fungi that were called the fungi imperfecti that couldn't reproduce sexually. That is an unreal grouping. Initially, fungi were categorized as plants, so they were in the kingdom Plantae, and the groups of fungi were then kingdoms. No, the kingdom was Plantae. They were phyla within the kingdom Plantae. Well, now fungus is the kingdom, but we still have phyla within that group. The other thing is, there are some organisms that some references categorize as protista that we are going to call fungi. In this class, if it looks like a fungus and it behaves like a fungus, we're going to call it a fungus. Also because the fungus name is applied to the protista-looking things. So what do you need to know? You need to be able to recognize the representative fungi. Again, you'll be able to identify them on site. You need to be able to describe the taxonomy as much as it's presented here, which is domain Eukarya, kingdom fungi, and then there are 4 phyla that you need to know. And then genus names and species names. So we're skipping class, order, family. You don't need to know that. You need to be able to name, recognize and name both sexual and asexual reproductive bodies for the fungi. Okay, the good news is there aren't that many examples. So domain Eukarya, kingdom fungi. The first phylum is called Mycota. When you see 2 O's and the second O has 2 little dots over it, I believe that's called a umlaut. And 2 dots means the O's are pronounced separately. So this is not pronounced oo, it's pronounced o-o. In here we tend to call it Mycota. Mycota. Anyway, if they're Mycota, their sexual spores are called oospores, and they're contained in a structure called an oogonium. Our examples are Saprolegnia, which is water mold, and Albugo, which is a type of rust.
Oh, can you go back to that slide prior, please?
Oops, I'm trying to go back. Oh, it did go back. Sorry.
I'm sorry.
Oh, now I went back too far. Was it that one?
Yes.
You're looking at the oogonium? It's in the lab manual.
Yeah, I like how you were on the screen.
Okay. Mycota. Yeah. Okay. It's in the other lab manual. So you have 2 examples here. Again, water mold. The reason this is called water mold is because it looks like a fungus, but it's in the water. Well, so some of the newer references are saying these aren't really fungi. We're going to call them fungi in here because the phylum, Mycota, is a fungus phylum. That ending doesn't fit in the protista category. There is a tube up here at the front that has water mold in it. Actually, it has water, and then it has some seed material, and what looks like a lot of kind of fuzzy stuff. It's the hyphae of the fungus. What you see on the slides, a goldfish that looks like it's growing threads off of it, those threads are hyphae. And this is an insect larvae, some type of dead insect larvae. Again, the threads are hyphae. It looks like a fungus. So we're calling it a fungus. It's in the water, it's water mold.
The fish is hyphae.
This is what our prepared slides look like. So this is Saprolegnia, water mold. These are the sexual oospores. What you see in the slide are a lot of hyphae in the background. You see the hyphae. And then you see these oogonia, and inside the oogonia are oospores. The illustration in the lab manual shows these little things that are called antheridia. Don't look for them. Never mind. They might exist, but you can't really see them. On the Saprolegnia slides, there's two regions of the slide. One region has the sexual otospores, and the other region has the asexual sporangiospores. So the sporangium is this sac. In this case, it's linear. These spores, when they're mature, will swim out. So this thing opens at this end, and the spores swim out. Because they produce flagellated spores, this is another reason why some references say, no, these are protozoa, not fungi. We're going to call them fungi. So again, that's Saprolegnia. You need to be able to recognize both sexual spores and asexual spores. Sexual spores are always named for the group, or I should say the group is named for the sexual spore. This next one is called white rust. That's the common name. Technical name is Albugoblytii. How many of you have ever seen rust on plants? So if you see berry vines, a lot of locally in the springtime, if you look at berry vines, what you'll see on the leaves, it appears as rusty colored powder. It looks like rust, but it's a fungus. And what you're seeing, the powder, is fungus spores. This one, the spores are white. This fungus produces haustoria, so those are the cells that penetrate into the plant cell. They absorb nutrients. So you're looking at the underneath side of a leaf, and what you see on the surface are these powdery patches. These are where the asexual spores are being released on the surface of the leaf. This is what our slide looks like. So this is showing Albugoblytii inside a plant leaf. You can't even see the hyphae of the fungus. Instead, what you can see are these oogonia that contain oospores. So those are the sexual spores. The rest of this is leaf. There's leaf epidermis, leaf parenchymal cells, over here a leaf vein. If you move back and forth on the slide, in another region of the slide, you can find this. So this is showing the leaf. The picture that I showed you where the leaf had the white spots on the bottom, the white spot is this thing. So this whole thing is like a pustule. This is the leaf epidermis, which is breaking away, and this thing is just full of spores, all of these spores. Because the spores were originally contained in a sac that was formed from the leaf epidermis, they are considered sporangiospores. Some references call them sporangioconidiospores. Don't go there. Okay, so we have in the phylum Oomycota, we have two examples, Saprolegnia and Albugo. They both produce sexual oospores and asexual sporangiospores in sacs, but the sacs don't look the same. Okay, the next phylum, we're still in the domain Eukarya, kingdom fungi, phylum Zygomycota. Here the sexual spores are called zygospores. So think about the sexual reproduction of fungi. You have to have plasmogamy, karyogamy, meiosis. When plasmogamy and karyogamy have occurred, you form a zygote. And in this case, the zygote is going to be visible. Our example is Rhizopus stolonifer. There's a picture of Rhizomucor in the lab manual, but we don't have prepared slides of it. So Rhizopus, sometimes called bread mold. You'll see this a lot on rotting vegetation. This is apple skins and kiwi skins. If you have a jack-o'-lantern, and a lot of times people leave them sitting out, you go back and look at it maybe several days after it was originally cut, and it'll be full of fuzz. That's probably Rhizopus. You can call it pumpkin fuzz. But anyway, this is what the sexual spores look like. So again, sexual reproduction involves two different fungi coming together. They undergo plasmogamy, karyogamy. They form this structure, which is the zygosporangium, and inside of that are zygospores, which you can't see. The zygospores form meiosis. These are the asexual spores. Again, they're in a sac. In this case, the sac is round. So if they're in a sac and they're asexual spores, they're sporangiospores. The nice part about our Rhizopus slides is usually both types of spores are the same color. But again, this is totally artificial. We have some slides where the rhizopus is stained green. Moving on to the next phylum, domain Eukarya, kingdom fungi. The next phylum is Ascomycota. Every time you have a phylum name that ends with mycota, the asco part, the zygo part, the oo part, that tells you what kind of sexual spores these guys make. So Ascomycota makes sexual spores called ascospores. They're held within a sac called an ascus. Sometimes the asci may be contained within a Cleistothecium. You don't have to recognize that on a lab exam. This is a big phylum. So the Ascomycota is a huge phylum. We have a number of examples. Saccharomyces cerevisiae, Claviceps purpurea, Penicillium, a whole bunch of species, Aspergillus, and Morchella. So here's what our prepared slide, we'll get to that. This is talking about what Saccharomyces is used for. Saccharomyces cerevisiae. Saccharomyces means sugar fungus, and cerevisiae refers to cerveza, and that's beer. So this is the sugar fungus used to make beer, or bread, same fungus. This is what our prepared slides look like. This is what the slide looked like when these slides were new. So they had cells reproducing with buds. The cells are pink, the buds are pink. So here's a bud, this is the big cell. There's another bud down here. And then you see these cells in the background that are blue. Those are asci with ascospores. Unfortunately, over time, those slides have all faded. So our prepared slides of Saccharomyces, everything is pink. You can't find those blue cells anymore, which is unfortunate. But now you know when they talk about this bud's for you, that's what they're talking about. Saccharomyces budding. You believe that? No. Well, anyway. This is another example. This is Morchella esculenta. So what we're looking at, this is a fleshy fungus. They do produce sexual ascospores in asci. So if you look at that, notice how that fungus has a lot of indentations in the cap. And where we find the spores are lining those indentations. So again, when you see a fleshy fungus, that is a reproductive body, strictly for reproduction. These are the asci with the ascospores. So in this case, the asci are linear. The ones of Saccharomyces were kind of oval shaped. Notice that we can determine this is sexual reproduction that led to the production of these spores because they're not all the same. Some are dark, some are light, some are red. So you've got genetic variation in here. So those are the sexual ascospores of Morchella. That's fungus on some green beans. Would you call it blue or green? Well, those are mycelia. So those are masses visible to the naked eye. They're made up of hyphae, and I think probably that's Penicillium. I left that plate with the green beans sitting on the front counter for a while until Sasha told me to throw it out. I was like, get those out of here. This is what Penicillium and Aspergillus look like when you're looking at the sexual ascospores. Penicillium, so these balls, these things are called Cleistothecium. This is a Cleistothecium of Penicillium, and around the outside you see what appears to be just kind of granular looking material. Those are actually hyphae, but they're very, very fine. They're difficult to see. This is Aspergillus. The Cleistothecium is significantly bigger. The spores inside are bigger, and you don't see any hyphae around the outside. So you have to be able to distinguish between these two. Again, the Aspergillus, the hyphae that you see around it are really minute, and you can't really see the ascospores clearly, whereas with Aspergillus, you can see the ascospores, distinct cells in asci. Those are the sexual spores, ascospores in Cleistothecia. These are the asexual spores. So Penicillium, again, these are conidiospores. The Penicillium spores are in line, more or less parallel chains, whereas Aspergillus, the chains are coming off from a central point in all directions, like a push ball. This is the orange. You find this in the center of the bag usually. There's two different images here. The one on the left, this is showing one spore. And then from that spore, this is a mass of hyphae that have grown out. This is one of the reasons fungi are so successful. If one spore lands in an environment where it can begin to take in nutrient, it will begin to grow and send these hyphae out. And then once the hyphae establish themselves, they're going to be taking in nutrients, and the next thing you know, your orange looks like this. This is mycelium. This blue colored and green colored, those are conidiospores. If you touch it, it comes off on your fingertips. If you see that green color on a chunk of cheese, you have to think, okay, that's aerial mycelium. What's in the cheese? Vegetative mycelium. Then do you chuck that cheese, or do you cut it off? Depends on how desperate you are to eat that cheese. This is Cladoceps purpurea. This is what we see in this Riker mound up here. So Cladoceps is a fungus that infects plants. And you see these odd-looking grains of the on the plant. You see the grains, but what they are is just a mass of fungi. The fungi have taken over this particular grain here. They cause a disease in plants called ergot. And if animals eat plants that have ergot, they get ergotism. And it's very bad for the animals. Cattle, here you see, if you don't know what an Angus cow looks like, you might not recognize the fact that there's something wrong with its ears. But their ears are usually quite a lot larger than that, and they're very hairy around the outside edge. This cow is missing the outside edge of its ears. And in the background, you can see one that has some damage along the back of it, and oftentimes it'll damage their feet. They tend to lose their extremities. This is what we're looking at on the prepared slide. This is showing a reproductive body where you're looking at a cross section through it. This is this thing. This grows out. So if this was the plant seed, you see these little things that look like tiny little mushrooms grow out. This little thing comes up. This is what it looks like when it's been cut. And these things are where we would find the ascospores. So they're also in sacs. They're not called Cleistothecia in this case. And you can't really see them. So here we see again, it's talking about this. The ascospores are in a perithecium, not a Cleistothecium. And then off to the side, it's talking about Cladoceps. Some of the material that is produced by Cladoceps can be used to make LSD. And there's been quite a lot of written information suggesting that people that participated in the Salem witch trials, that is, the people that were accused of being witches, they were being accused by people that had been eating contaminated rye grain, and if the grain was contaminated with Cladoceps purpurea, these guys caused hallucination. So people were actually seeing their neighbors fly, but they weren't really flying. The people were eating contaminated grain. This apparently is somewhat controversial because there's been other writings saying, no, that's not really what was going on. But these guys do, they are potentially hallucinogenic. So now we're moving on. Domain Eukarya, kingdom fungi. Our last phylum is the phylum Basidiomycota. If they're in the phylum Basidiomycota, it's because they produce sexual spores called basidiospores, and they're supported by a little structure called a basidium, which kind of looks like a little tiny cow's udder. Our example is Cladoceps, our example is Coprinus comatus, called the shaggy mane mushroom. They look like this. Also, they're sometimes called inky cap mushrooms. As the cap begins to dissolve, you see this black stuff running down the stem, and it's spores. These spores are usually released, sometimes they're released into the air, but these tend to be carried by water. Anyway, what you're looking at is the underneath side of the cap, and these are gills. And on the surface of the gill, you see these Basidiospores. You can't really see the Basidium. So when you go to Safeway or Ray's or wherever, if you buy mushrooms, and if you look at the underneath side, you see those little, look like spokes of a wheel. Those are the gills. And every single one of those things is coated with Basidiospores. So again, that's it's a sexual reproductive body if you buy a mushroom. And it has a tremendous reproductive potential because each one of those things can grow a new fungus. Now these are slime molds, and you don't have to know the slime molds. And unfortunately, we don't have any slime mold examples. But if you wanted to know about them, there's two categories of cellular slime molds and acellular slime molds. And you don't need to know them. They come in a variety of colors. Sometimes people have slime mold races, and they set them up in containers where they have rows of oatmeal, and they grow down the oatmeal. All right, so I'm going to put this information on the board. It's also in the lab manual. And you may continue to do whatever it was you were doing before I did it. Or you can look.
Sorry guys. My bad.
And I'll put this thing on the website.
I saw that.